Now the P site is full with this tRNA and the A site is empty. What happens is that the A site can now take another tRNA. We have another codon, nucleotides number 4, 5, and 6 have another codon, a different sequence, and they have a specific tRNA that can bind to them with complementary anticodon, and it carries the specific amino acid. Now, when this happens, the initiator tRNA is going to leave the P site, and once it does, the bond that was formed between the amino acid and the tRNA breaks, and this liberates energy, it releases energy, and this energy is used to create a bond between methionine and the second amino acid, and this is what we call a peptide bond, the bond between two amino acids. So now we have methionine left the P site, and the ribosome moved forward. So now the second codon is actually in place of the first codon. The second tRNA is now in the P site, and the A site is empty for a third tRNA to come and bind to the third codon, carrying a third amino acid. So now the same thing is going to happen. Now the second tRNA is in the P site. It is going to leave, and it is going to break the bond it has with the second amino acid. And once it does that, it releases energy. This energy is used to create the second peptide bond between the second and the third amino acids, and the ribosome moves forward. So now the third tRNA is in the P site and the A site is empty again. So the fourth tRNA can come to the A site and bind to the fourth codon, and with it, it carries the fourth amino acid, and then the ribosome moves forward, releasing the tRNA in the P site and forming a peptide bond between the amino acids, leaving space in the A site for a different tRNA to come and join. And this is what happens. This is elongation. This keeps happening until the ribosome reaches a specific codon called the stop codon. The stop codon tells the ribosome to dissociate, to leave the mRNA, and stop translation. This is the final step of translation. It is called termination. And this is how translation stops. And once translation stops, the ribosome, the two subunits, are going to dissociate from the mRNA and from each other. And we will have a polypeptide strand. It is made of many amino acids that are bound together by peptide bonds. And this is how we translated the mRNA into protein, actually. This code is universal because in all organisms, these codons code for the specific amino acids, and these codons are coded for by genes. So this is why we call DNA the genetic code, and it is universal in all organisms. The genes will code for the same amino acids, so the same proteins.